Good day, everyone. My name is Patrick Wheatle, and the topic for my final presentation is the genes involved in atopic dermatitis. So, before we really get into the genes that are involved, we need to understand what atopic dermatitis is. So, according to the National Institute of Arthritis and Musculoskeletal and Skin Diseases, or NIMS for short, atopic dermatitis, or eczema, is this chronic, long lasting disease. It can cause inflammation, redness, irritation of the skin, and other symptoms can include cracked skin, small fluid filled bumps, and lichenification, which is this thickened, leathery skin from chronic scratching. So, look at the picture right here. So, this is actually my hand, and I have this lichenification because I scratch my skin a lot, which I really should not be doing. And if you have eczema, please don't do this, by the way. So there are three things that I want you guys to learn from this presentation. So the first is that atopic dermatitis is a complex and multifactorial disease which involves immune system dysregulation and dysfunction of the skin barrier. Atopic dermatitis, it results from a feedback loop. So let me give you an example. Genetics, it can cause a weak skin barrier, and a weak skin barrier can cause increased allergen or microbe entry, and increased immune activation causes inflammation, and increased inflammation, it can cause further damage of the skin barrier. The second thing is that genetic susceptibility, it drives disease risks. So atopic dermatitis is polygenic, which means that multiple genes, it contributes to its development, like the FLG gene, which affects the skin's protective barrier. And the third thing that I want you to know is genome-wide association studies have advanced the understanding. So we have the studies including the Chinese Han and Japanese groups in the population. So the findings, it shows that atopic dermatitis risk, it involves multiple genetic pathways, and that certain risk alleles, it can vary among ethnic populations. And the goal of this presentation is to demonstrate how atopic dermatitis is strongly influenced by genetic susceptibility. As for my methods, I used a research-based approach. So the first, it was topic selection. So I focused on the genetic basis of atopic dermatitis, especially susceptibility loci. Next was database search. I used sources like PubMed and Google Scholar, and I looked up terms like FLG mutations and genome-wide association studies. Then I did screening and selection. I narrowed it down to the most relevant studies, including research on Chinese and Japanese populations, and studies that looked at both immune and skin barrier factors. After that was data extraction. I pulled out key findings like important genes and differences between populations, and I compared the results between them. And finally, is synthesis and analysis. I combined everything to understand how genetics influences atopic dermatitis and how this can improve patient care as well. And here are my results. So the first result was that major susceptibility locus was found on chromosome 3q21. So a study by Lee and his colleagues in 2000, they found a susceptibility locus for atopic dermatitis on that chromosome region. And it corresponds with the COL29A1 gene, which encodes collagen type 29. And that's a protein that's important for maintaining skin barrier integrity. This collagen is normally found in the outer layer of the skin, and it helps protect against environmental irritants and allergens. But in individuals with atopic dermatitis, the COL29A1 expression is reduced or it's absent. Later research by Soderhall and his colleagues in 2007, they found out that genetic variants in the COL29A1 are associated with an increased risk for developing the condition. And result number 2 is that 2 susceptibility loci were found in the Chinese Han population. And a GWAS study by Sun and his colleagues in 2011, they found the loci associated with it as 5q22.1 and 20q13.33. But they narrowed it down. They looked at the 5q22.1, and it was later identified to have the genes transmembrane protein 232 and solute carrier family 25 member 46. And we're going to focus on the transmembrane protein 232 because it promoted the inflammatory response for atopic dermatitis, and that's why it was the primary candidate. And as for result number 3, 8 susceptibility loci were found in the Japanese population. So a GWAS study by Hirohita and his colleagues in 2012, they found 8 new susceptibility loci associated with atopic dermatitis. So the first one is very long. It's IL-1, RL1, IL-18R1, IL-18RAP, major histocompatibility complex, NLRP10, GLB1, INPP5D, KRAK, OVOL1, and ACTL7A. So Psalm 139 verse 14 says, I will praise thee, for I am fearfully and wonderfully made. Marvelous are thy works, and that my soul knoweth right well. So we should still be praising God even though all these genetic diseases that causes a lot of pain and suffering are in this world. So I chose active listening as an important soft skill because it helps a patient feel heard. So a study by Kawamichi and their colleagues in 2014 revealed that active listening, it actually activates the brain's reward system and helps people evaluate experiences more positively. That's a really good thing. So to conclude it off, atopic dermatitis is a very complex disease. Genome-wide association studies help to advance its understanding, and that active listening helps atopic dermatitis patients feel heard. So this marks the end of my presentation. So I would like to thank Dr. Richardson, Dr. Vanderpool, and my classmates for helping and critiquing my research presentation. And here are my references. Thank you for listening.